All right. All right, welcome everybody to another StaPH-B call. Today I'm pretty excited that we have Danny Park here speaking with us. Danny is from the Broad Institute, working with MIT and Harvard, and also doing bioinformatics consulting work with the Massachusetts Public Health Lab, who's also the Northeast Bioinformatics Regional Resource. And today we're going to be talking in general about workflow managers, and then Danny's also going to get specifically into the Cromwell system and the workflow development language that is being utilized by both Broad and also, my understanding, is the Massachusetts Public Health Lab, or at least at the beginning of integrating those technologies. And it really feels like this is going to be the next step in public health bioinformatics in terms of all of us being able to efficiently use our compute resources specifically. So very excited with that. Danny, from there, if you want to go ahead and share your screen, and I'll go ahead and mute myself, and we'll take it from there.
Sure. Here we go. Let me share my screen. So I hear that there's been some excitement and a lot of looking forward to discussing this topic on a StaPH-B call. I will have some slides, and I'll tell you some of what I have learned and just kind of what I'm currently doing in this area. But for me, actually, I kind of want to wrap up some of this as quickly as possible. Don't you love it when it auto advances? Maybe I'll actually quit the slideshow thing and just do this instead, because why not? Yeah, because for me, I'm actually looking most forward to just the discussion time. I hear some people have been dabbling with a couple of different languages, and I just want to hear how that's been going so far, what people like about whatever they've been playing with, and that type of stuff. So I'm actually quite curious about the part after I stop talking. So let's see here. Today, I will discuss a little bit about kind of what pipelines are, why we're even talking about them, that kind of stuff. I'll talk about languages, and orthogonal to that, but also highly intertwined sometimes, are how you execute those languages. In some cases, they're very tied together, but sometimes they're, in an ideal world, they're quite separate.
Is the screen blacked out for everyone else?
I will mostly talk.
Is the screen blacked out?
Yeah, it does look. Yeah.
Okay, why don't I switch to sharing single window or something like that?
We had such a flawless technical check.
Well, I know. It was so brief. Can you still hear me though? Just checking.
Yes, loud and clear.
Okay, all right. Let me switch back to, I'll share this one window, and then I'll switch which window I'm sharing when I go to, all right, so I'll just do this one.
Did we lose Danny?
I felt like I got lost for a second. Am I back?
You are back, though we aren't seeing anything on the shared screen. Oh, okay. There it goes.
There we go. I got something.
Perfect. We're on the slide. We'll edit this in post.
There's always something. Okay. So the one caveat I was going to say is I will focus on the languages and the platforms I'm most familiar with. And I will try my best, because I think people are interested, and also talking about some of the other popular ones out there as well. I won't be able to speak authoritatively on with experience on some of them, but I can, from a high level, what I understand, I'll also talk about how they're similar or different. But I'll try to walk through what it looks like in the languages that I'm most familiar with. Okay. So why do we, or what do I mean by computational pipeline? So I'm thinking about, it's one thing to have like, I figured out how to Dockerize a particular tool and I run that thing many times on different types of data. But for many of us, we work with certain pipelines, a pipeline being, something just conceptually where you're doing multiple computational steps. And of course you could glue them all together, and that's usually the first thing we do, especially if it's a simple glue together. But sometimes those steps are thought of kind of separately by the user. They're conceptually different things. Like I might align reads, I might assemble reads, I might do all sorts of, like annotate the genes or whatever. And those are steps that someone who's doing analysis might want to turn knobs separately. They might want to swap in a different program. I got my favorite program for assembly. I want to do that instead. More importantly, they might want to actually QC certain steps in between. So you might actually care about some of those intermediate files that kind of come through. So I'm usually not talking about steps like, oh, let's convert a FASTQ to a BAM. That's more like nothing really happened to data there. That's just format conversion. I'm talking more about like you did something that legitimately took some compute because you're doing some sort of algorithm on something, right? The other thing, reason why we might want to represent them separately instead of just having a monolithic Docker package that has a script that runs steps A through D all at once, is that some of those steps might require like different types of hardware. Maybe something needs a lot of memory, maybe, and you might want to actually think about them differently. If you're working on the cloud, you might spin up a different kind of VM for a certain step. If you're working on a cluster, you might allocate more like job slots to a certain step if it has more resource requirement, versus other lighter steps where you could run like 100 of them at the same time. There's many ways to do this. Like I said, you can just glue everything together in a bash script if you wanted to on a single Docker image. But part of the reason that much of the field has been moving towards these kinds of formalized pipeline languages, and which I think is a good thing, is obviously for reproducibility, so that you can run it again in 10 years or something. But the big one is so that we can share this stuff, right? In an ideal world, if I've got a pipeline for X, and how many times have you had this kind of discussion like, oh, does anyone have a pipeline for calling AMR genes out of a short read data set? Like, oh yeah, I got one over here. Here, have mine. It's not quite so simple. Like you wish it was. You wish you could just say, here's my pipeline, run it. Oh yeah, I'll just run it on the same thing that I run all pipelines on. And we all have something like that, even though our setups are different. Having some sort of formalized language helps kind of have something a bit more shareable in that. Funny thing about this view is that you're going to get this a sneak peek at all the other slides I'm about to to go through. All right, so there's two main things as you think about genomic, like kind of bioinformatic pipeline, like implementing something like that more formally in your context. If you decide that you do want to go more formal, you want to decide what language do you want to use. There's a few different ones out there, and what kind of platform or executor, or like how are you going to run that language in your lab. The two, it'd be great if they were very entirely separate things. The language is basically who you can, the people who speak the same language can share information together, right? So if we're all speaking WDL, then we can all pass our pipelines around. If we're all speaking Nextflow, then we can all pass our pipelines around. Ideally, these would be two very separate choices, but they are interlinked because not every engine, supports every language, and so obviously there's some choices there. Within a certain language, some of the choices on what type of platform or execution engine you use may be defined by the usual things that constrain your compute, right? Like you have a certain infrastructure, are you allowed to use this type of a cloud vendor or not, or that kind of thing. But I think if this is something that your lab is starting to move in towards, I think my recommendation is try to just think about how to maximize the portability factor. Whatever you set up for you, do you see how this would work in terms of sharing it with everybody else? So over time, things that approaches to this that I've seen before. So when I first joined the Broad like 15 years ago, back before there were kind of like standardized language for it, basically what everyone would do is like build their own in-house thing. So we had this humongous, very thick, kind of Java, SQL, whatever thing that managed our cluster and our compute jobs for us. I'm sure lots of other big institutions had that type of stuff. It's not really maintainable. It's not portable. You can't take a pipeline there and run it anywhere else. It was a necessity of the time, but increasingly it's not something that I would recommend these days. I've also moved away from things that are kind of uncontainerized, and by that I mean I've come to realize over the years that like Conda just by itself doesn't quite solve the problems, because every once in a while they rebuild like everything and your whole thing breaks. And so it works great to set up a Docker image. I use it all the time in my Docker files because it's just quick and easy. And then once you build your Docker image, it snapshots it, and like all of Bioconda can go down. Who cares? Your Docker image still works, right? So that's kind of a one, like relying on that alone is something I've kind of moved away from. I do have a whole slide just about how, and I don't want to like kind of hammer this too much. I'll simply say that SnakeMake has been wonderful for our group in the period of time that we've used it. We started using it probably around 2014. It was it's definitely one of the fastest ways to get started with throwing together some pipelines. Definitely recommend it for folks who just kind of need like a rapid on-ramp to something. And in 2014, it was probably, a lot of the other options weren't that mature yet. But I've increasingly run into some like just very fundamental limitations of it. And these aren't things that can be like fixed in future versions. These are almost like in there almost by design. One of the problems I run into is that second bullet is, if you've played with SnakeMake before, make-like things, they construct these, the whole workflow kind of backwards from how most languages kind of construct it. So most workflow languages will say, okay, here's step A, step B, step C. Step A takes this input, produces the output. And it kind of, you describe a workflow by pushing data from the beginning to the end. And make, and SnakeMake as a result, is kind of backwards. You talk about what you want, like the end product, and then it kind of looks at what you got and kind of infers and works backwards. Pretty cool. It's really good for kind of reuse of, not overdoing the work if it doesn't have to. But it actually means that it's like impossible to express certain types of steps, like, this one step produces output that I don't know what the names of the files are going to be, or even how many output files I'll have, until you actually do some compute. And the way people get around that is you set up some interesting input rules in Python. And that brings me to the other kind of bullet point here, which is the flexibility of SnakeMake to just inject Python everywhere makes it very quickly devolve into something that's hard to read and maintain, and also hard to port to some other language. So it's also very clear that it was kind of built for like a single machine, or in some ways it can kind of be finagled to work on a cluster. I did a lot of muscle work to get it to work on our cluster here. And it can kind of work on the cloud, but it's really kind of, it's best when it runs on a more mainframe kind of mode of operation, like one big machine everyone's sharing. So that's something I've slowly been moving away from, though it's definitely served us quite well. So here are the kind of, if you were to start from scratch and say, where would I get started? These are the three main ones that I think are kind of, seem to have the most popularity in the genomics world. There are other ways of doing this outside of kind of computational biology and genomics, but these seem to be the most common in genomics. I will speak mostly about WDL, which is the workflow description language, because that's what I have experience with. CWL is quite popular as well, and Nextflow in certain circles. I say this with a little bit of a disclaimer in that I work at the Broad, and WDL was made by the Broad. That being said, I was kind of a skeptic at first. It's made by a different group. I'm in an entirely different, kind of space. And when I was initially shopping around for languages, I actually didn't, I wasn't convinced on it until I saw certain industry partners of ours really move in that direction, and I'll talk about that later. I actually think pipeline languages for genomics are an excellent example of convergent evolution. They're all born around the same time because everyone independently discovered a need and then started working on it themselves, not realizing that other people were working on it. And they all come up with very similar but slightly different solutions. And so that's where we're at today. And that's why these types of things exist. If you ever wonder, why don't they just use this one? It's like, well, they all kind of came up around the same time. CWL is interesting because it was actually born straight from the get-go out of a kind of a consortium of industry and academic partners. If you say a little bit about kind of just like who owns them and runs them today, the first two, WDL and CWL, tend to be more, in certain circles, more first-class citizens of kind of like within certain standards bodies like the GA4GH, and I'll talk a little bit about GA4GH a little bit later. But Nextflow is so close in terms of how it works that it's essentially like GA4GH adjacent. So a lot of tools that support one of these two will often also support the third. But you'll also see some places where the Venn diagram doesn't quite overlap. In terms of who actually runs these, most of these are actually run by these kinds of open consortium processes. There's like formal governance structures where lots of different entities are contributing to it. CWL was born that way. WDL started at the Broad, but quickly became that way once it was realized it kind of had to become that way. I'm not as familiar with how Nextflow is, as a language, is maintained these days. So a lot of what I am going to say about it is a little bit just kind of off of some Google web searches and stuff. So correct me if I'm wrong there. They all have their adherents. They're all quite popular in different circles. They all have different entities that are active contributors to producing pipelines in those languages. There's places where they tend to get stored and distributed and stuff like that. So this is what I mean by ecosystem. When you pick a language, you're kind of picking an ecosystem. Luckily, if you're in any one of these three, because they're kind of semantically not that different, if you don't have a particularly complex workflow, you can easily write them multilingual if you really wanted to, and some people do. So sometimes that's the best answer, actually. I'm just kind of like, sorry, going through a little bit of at least what I know of just kind of similarities and differences between these different languages. One of the choices I said you're going to have to make is how are you going to execute any one of these languages in your setup? How are you going to run a pipeline, right? And they have different answers to that question. They usually have their kind of their flagship tool that's used to run stuff, but some of them actually kind of exist in ecosystems of a lot of interoperability by design. The WDL language, its primary implementation is an engine called Cromwell, and I'll talk a little bit about that more later. Cromwell is a software package you can download and use to run WDLs. You can set it up in different ways to run it in your setup, like say you can run it on your compute cluster or your big compute server, or you can use it to spin out jobs to the cloud. But there are like execution engines, but there's also platforms, especially for the first two, WDL and CWL. There are cloud platforms that adopt these. So Terra is a cloud platform that's based on Google that is run by the Broad, that under the hood has Cromwell just running all its stuff. This is to say, if you don't want to run one of these types of schedulers yourself, you can just go online, set up an account, and pay someone to run it for you. Similarly, DNA Nexus is probably the oldest genomic cloud compute company out there. I think they're at least 15 years old. Seven Bridges is also almost as old. So these are commercial companies that you can just set up an account with, pay them money, to do genomic computes and stuff. And usually their business model, they have highly tailored projects, but both of these companies have give you the ability to ingest your own WDLs or CWLs or something like that. So Seven Bridges, DNA Nexus are both commercial companies. Many of you are familiar with the Galaxy project. This is probably one of the oldest academic projects in this type of space of giving a nice kind of web GUI for like kind of build your own little portal for spinning up jobs and managing a cluster. The Galaxy project was one of the earliest kind of people involved in the CWL project. I think CWL language supporting Galaxies, again, I'm not speaking from direct experience, but just from people I've talked to, it's not quite a first-class citizen there yet. You can't just like, but it's something that's it's moving in that direction. So I put a question mark there. I don't think it's quite so easy to just run CWLs in Galaxy, but I think that is the idea. The Broad's Cromwell software engine can also run CWLs. There is another tool called Toil that runs both WDLs and CWLs. There's a new tool called Mini WDL from CZI that runs WDLs. And I think, I'm not sure, and please correct me if I'm wrong, that the way to run Nextflows is using Nextflow's tools. I don't know if there's other kinds of corporate, like, you know, commercial platforms or competing implementations of software for it. If this is just, you use the Nextflow engine to run your Nextflows. So anyway, okay, so the language itself, kind of going back to the language, this isn't terribly that important, but it may just kind of get a little bit to your level of comfort with getting started. But it also says a little bit about how some of these things kind of behave in some of the edge cases. So WDL is the only language here that was it was written as a brand new language. So in order to make it work, they wrote a whole ton of like parsers and stuff. So if you ever like, you know, if you major in CS in college, you probably learn about like languages and parsers and lexers and all this kind of stuff. They did all that stuff. And what that means is when you look at a WDL file, you can't break the rules. Like you just have to follow the language, right? Whereas a lot of these are built on top of pre-existing languages. So you can like, there's YAML and then there's CWL, which is like kind of a subset of it, right? But sometimes you can throw in some other YAML in there too. Groovy is a kind of Java-like language, and sometimes you can slip in a little Groovy like Java type stuff into Nextflow. This is kind of what I was saying is like my main gripe with snake-make files is a lot of times like actual Python slips into there and it gets actually, you can't just write a parser to turn that into another language. Okay. Let's see. Okay, so I said I was just going to show and tell a little bit about what my pipelines look like. My pipelines aren't special. You've got pipelines, I've got pipelines. I mostly focus on viral genomics, so I've got some stuff for metagenomics. You know, I run Kraken and that kind of stuff. I assemble and align genomes, pretty normal stuff. At the end of the day, you probably don't care so much about what's going on in the guts of each step, just how do I thread it all together, right? So for me, part of the reason I ended up enjoying porting my stuff into WDL, well, part of what motivated it, we had originally had a lot of our stuff years ago. Some of our stuff was kind of co-implemented in DNA Nexus's old kind of proprietary language format, just out of an old partnership we had with them. And one of the things that I thought was interesting was when I saw them switch over to kind of embracing both WDL and CWL. So what they ended up doing is writing essentially compilers that convert those languages into their native format. That actually made it a lot easier for me, because then I could actually express my pipelines in a more kind of standard format that now were portable. So for me, I converted to WDL to have a single kind of bit of code that describes my pipelines that can run both at the Broad and in DNA Nexus and on Google and all this kind of stuff. So that was my motivation. One of the things I enjoyed about it was that it's actually quite elegant once you learn the language. This little snippet of code is this pipeline. So if this pipeline is conceptually easy to understand just because it's icons and arrows and stuff, it's actually just these 5 steps. Call this, call that, call that, call that. And you can see how it's chaining. Oh, the input of this is the output of that, and so forth, right? So obviously there's complexity under the hood. We hide it up here in these imports, right? Each of these steps, there's much more lines of code, but the big picture is easy to see at the high level. So that's kind of one of the things I ended up enjoying about it. And so now that I've got my pipeline written up in a WDL, I can now run this on DNA Nexus's commercial platform. That, by the way, is historically just a big layer on top of AWS. So under the hood it's AWS, and recently they started also doing it on Azure as well. So basically that means I can run it on Amazon or Microsoft. I can run it on the Broad's Terra platform. I'll show you both of these. I can just DIY my own execution engine. So I can grab Cromwell, which is what Terra uses under the hood, and run it on my cluster at the Broad. I can, spin it out to Amazon or Google myself, or actually recently Microsoft as well. Or I can just run it on a big box or on my laptop if the hardware permits, right? So that's kind of like the portability dream. So any questions? I was going to do a little show and tell of just and kind of high level of what the code looks like and what the execution looks like. But any questions so far?
Hey, Danny, I had a question, especially about WDL, and then really appreciate the history of all those workflow languages. I like the term conversion evolution there. But in terms of WDL and these other workflow languages, I read about different workflow managers, and I've had almost been overwhelmed by the different options that we have here. But were things like WDL and CWL and Nextwell, were those bioinformatics specific workflow languages?
I think all of these, and for anyone who has experience with these will kind of see, the way that they're written doesn't have to be anything specific to bio or genomics. They were written to solve, bioinformatic and genomic problems. So they were addressing that particular need. There's no particular reason why, you couldn't mine Bitcoin with it or something. So.
Okay, I got that. And then I also had a question, actually 2 more questions. In terms of those importing, so if you go back to that last slide and you were showing how you can import what's actually being called, whenever you're doing something like taxon filter to taxon, are you importing WDL executables? So when you're actually, for example, calling the Docker images to run the analysis module, those are all written in WDL. It's not like you're bringing in a Python script or a Bash script or something like this.
I think it might be best if I show and tell what the code looks like.
Okay.
That might answer your question.
Yeah, definitely.
See under the hood.
Yep.
Yes.
Because I
did too. Thank you.
some of it is a little abstract until you actually. That's kind of why I wanted to show and tell a little bit there. Okay, so let me now switch to actually. Let me see if I can go back to sharing the whole screen again. It choked a little bit when I did that last time, but let's try to do that. Okay, so where was it? Okay, now. I thought I had the. Oh, you know what? I think I was just supposed to click this. Let me know if last time I shared my whole desktop, I ran into some connectivity issues. So, but you can see
it looks good.
My GitHub. Yeah.
Yes, though a bit small.
You know what? Maybe just blow it up a little bit. Yeah.
Looks good.
Okay, let me show you the thing that I just had on there, which was this particular thing, which I think was just this, this whittle. Okay, so as an example, let's take a look at any one of these steps. It probably doesn't really matter, right? Okay, so yeah, like filter to taxon, for example, right? Okay, so this is something that has 5 things. We call step 1. Step 2, the input requires the output of step 1. So Cromwell, or whoever's executing the whittle, automatically understands, oh, I can't run step 2 until step 1 finishes. So it creates a little DAG, right? Interestingly enough, if this was not the case, it would actually run both steps simultaneously because it knows that they don't depend on each other. But because step 2 depends on step 1, it creates a little chain where, you know, it runs one and then it runs the next. Similarly, step 3, you can see this input is asking for the output of this step. So, you know, so on and so forth. That's actually the case for all of these. Each of these require, and some of these actually reach back a couple steps for their inputs. If you're wondering what's under the hood in some of these things, so let's take a look. So that's called a workflow. A workflow calls these tasks. Now, if I actually look into the tasks, here's the meat of it. The task is, and this is much more complex than a simple, partly because I guess I'm not showing you the simplest one, but it's evolved this kind of complexity over time in order to simplify what it looks like on the high level and also kind of expose knobs and that type of stuff. But if you look at some of the simple hello world examples or GATK snip calling, like those tend to be much simpler files, but we'll just start from here. The task is the atomic compute step. And basically, the way to think about it is a lot of this is kind of thought of from the cloud computing context. It translates just fine when you're running it on a cluster. It's a lot easier to take a cloud computing paradigm and stick it on a cluster or on a mainframe than it is to go the other way, because the other way usually runs into problems like, where's my file and all that kind of stuff, right? But if you look at this language, think of it in a cloud computing context. Basically, a task is when you run it, it's going to spin up a VM somewhere in the cloud. It's going to pull down a Docker image. It's going to run a script. And before it runs the script, it's going to pull down some files from cloud buckets onto that VM. And then after your execution is done, it's going to take outputs that you said were outputs, and then it's going to push them back to buckets, right? So this is the cloud kind of computing paradigm. In the case of running it not in the cloud, that's totally fine because you can do all that orchestration anyway. It's absolutely necessary for the cloud. It's kind of optional for not cloud, right? So what you see in the task is on the top, these are the declared inputs. So it's basically saying when you spin up this VM, these files, you're going to have to go find them in a bucket somewhere, pull it down, and bring it onto the actual VM before you do anything else, right? And I'm not going to go into details here, but some of these things are optional parameters, and some of them are mandatory, and yada, yada. There's knobs you can turn if you want to. This is our way of providing the ability to do some complexity if you want, but by default, not really requiring it. Before I get into that section, when I come down here, after your whole thing is done, here, then if the job was successful, you're going to look in these little file paths, find some outputs, and spit those back out onto the cloud. But this little runtime section is kind of the kicker here. This basically says, take this Docker image, which we made a little more complicated than necessary by making it a variable. So if you go up here to the variables, this little variable here, this is my Docker image that I use for this step. You can find it here at this, Quay repository. It could be on Docker Hub or whatever, right? You say, I want to run this Docker image on a VM that has this much memory, this much CPUs, this much disk, right? And it will spin up a VM that has this much memory, this much disk, this much CPU. It'll pull down the Docker image. And then what is it going to run? Well, it's going to run this script that you write in the command section. Now, most people's bioinformatic containers declare bash to be the entry point. So basically, this section here is bash, right? This is like a little shell script. So it basically says, given an input that comes down somewhere, run this little script. And these are all commands that exist in my Docker image, right? So this little Python script is in the Docker image. All these little variables are, SAM tools, whatever, fastqc, it's in the Docker image. So if you want access to some sort of software of a particular version, you provide it in this runtime, the Docker image that you want to run, right? And then if you want to do some a little cleanup or run this or run that or some other stuff, then that's what you put in this little script section. I say this is shell script because the Docker image's entry point is shell. But if the Docker image's entry point was like R or MATLAB, then this could all just be R or MATLAB code. So nobody I know does that, but if you if you wanted to add a little pain, you could. And basically, this is my description. The goal here is to be very generic and reusable and portable. Just to explain what this is a step that takes a pile of reads in an unmapped BAM file. Most people will take a pair of FASTQs, but I think our group is just kind of, don't worry about that detail. Just a bunch of reads, and it'll align it to the human genome, either with BLAST or BWA or BMTag or whatever you want. It depends on whatever optional input files people provide. Give me an index database to align this to, and then it throws away all the hits, right? So that what you have afterwards is depleted from whatever matched this database, right? Now, we default these values to be like the human genome, so that basically we do a metagenomic sequencing run of some pathogen, like a clinical specimen on a MySeq. We run it through this step. Now I got something I can share on SRA because it's not filtered down to anything, it's filtered away from human stuff, right? So that's the first thing we do. There's other steps here like filter down to something and yada yada. You can imagine these little kind of Swiss Army knife type steps that if you glue it together in a pipeline would be really useful. So basically each step that would be really useful, we kind of code up as a task. And then gluing it together in various ways, that's the workflow. Does that make sense? I realized there was a question on the chat. I think I missed it.
Yeah, that made a lot of sense. Thanks for showing us that. I think my favorite part of the WDL thing there is actually that output formatting and then retrieving it per call is in an almost like object-oriented fashion.
Yeah, what's interesting is in the cloud paradigm, right? You spin up a VM, you do some stuff on it, and then you throw it away. The whole machine gets thrown away and anything on there gets thrown away. So say you have some program that you ran from somebody else that somebody else wrote and it's kind of sloppy and it spills all sorts of temp files all over. It doesn't matter because it all gets thrown away. The only things that get saved after the execution is done is anything that you explicitly wanted to save. I want this file. It's going to be called this, such and such.clean.bam. Oh, what's that? Well, this is a variable from up above, right? So, you could say it's, again, there's some complexity here, but it could just be a fixed string, right? So if you see that file, and you can see what I'm doing here is like, okay, well, there's SAM tools, like this produces this output file, and that's how it's producing that file name. So if your shell script produces a particular file name that you can predict here, then you say, I want to save that, and then it's going to put it in a bucket and that kind of stuff. So
great, thanks, Danny.
Go ahead.
It didn't look like there was questions on the chat, just Robert noting some of the commercial use of Nextflow, and also Scott mentioning how some of the international foodborne labs using Innuendo, which sits on top of Nextflow.
Yeah, out of the yep. No, that's great, especially since I lack some of the background there. Okay, so that's what some of the code looks like. There's plenty of other different workflows that we kind of play around with here. Some of them are more complex than that. I showed you a simple linear workflow, step 1, step 2, step 3. You can also do scatters and gathers. I'm not gonna, actually, want to just show it to you real quick. I'm not gonna like explain it too much, but here's a workflow that I may have mentioned before that we tend to do all of our demultiplexing and base calling in the cloud mostly because of reproducibility. Like we just want to be able to do it again later and not rely on what the MySeq produced. So basically, our first step a lot of times is you take a zip up directory of the whole BCL directory off the MySeq, and then this task spits out a whole ton of little, reads, right? Pairs of FASTQs, or in our case, unaligned BAM files, whatever. Then you can scatter on those, and basically this little section here calls a bunch of these other tasks on every single file that it saw coming out of that. So if my flow cell had 20 samples on it, now it's going to parallelize 20 times and call all these things in parallel. And you can do gathers after the scatters. So after that's all done, then take all of those things and maybe produce some summary reports and aggregate statistics and so forth. So not going into the details, but you can kind of see how it can quickly get complex. You can have branching logic and stuff like that too. And that is true for all three of the major kind of languages that I would recommend people consider. Okay, so that's what the code looks like. Why don't we look a little bit about what it looks like to execute and see the execution or see the output of these types of things. So one of the easiest ways that I can kind of show you this, sorry, it keeps, DNA Nexus is one of those platforms that logs you out like all the time. So I gotta like keep logging back in every 15 minutes. I'm sure we all enjoy doing those things. Let me get my little code. I'm just showing it to you this way because I think it's easier to visualize, and I think it's easier to, even if you'll never use DNA Nexus or whatever, I think it's easier to understand how it's consuming the code that you just saw in GitHub, all right? When I show you the Terra side, you'll kind of see hints of what Cromwell is also doing under the hood.
Danny, can you zoom in again and maybe full screen that window?
Yeah, of course. If you want to go here, because I ran something here recently. Okay, so DNA Nexus is a web platform. You pay them money, they compute stuff for you. That's kind of the idea. You throw your data on there, you can compute stuff. Anytime you want to compute stuff, they just rent machines from Amazon to get it done. And that pipeline I had just shown you in GitHub, let me just show you what it looks like here. This is the assembled de novo with complete pipeline. If I click on it here in DNA Nexus, you kind of see their web UI job launcher, which is kind of nice and kind of slick, but you can kind of see how it interprets the WDL. This comes from the WDL. So basically that WDL runs through a little compiler that turns it into the thing I just clicked on in that DNA Nexus project. And you can see, I mean, they add on a little bit of boilerplate. So this top line you can ignore, and down at the bottom, there's some final outputs line you can ignore. But these 5 steps, deplete taxa, filter taxon, assemble scaffold, this is the WDL, right? In fact, all of the required input files come from that input section of the WDL. The output files are the output section of the WDL. Notice how the output of deplete taxa is the input to filter to taxon. That's because the workflow WDL joined the two together, right? And so the way DNA Nexus works is you fill in some of these MISID requiring inputs, and then you hit run as analysis, and then it starts executing that WDL in its platform. This shows you how to fill in some of the file inputs. It has an interface for inputs. See, some of the inputs were strings or integers or that kind of thing. Do you remember this gobbledygook? That was one of the default values for one of the optional parameters, but yeah, that was out of the WDL. So that's kind of DNA Nexus consuming or being able to take that WDL. And when you actually execute it, I'm not going to fill that out, but I'll show you what I ran yesterday. When you actually execute it, it goes into this page of kind of the history log of all the things that you've executed. And by the way, this project is just someone in my group was. I mean, there's been a lot of coronavirus genomes on GenBank and GISID and stuff, but he noticed there's actually some short reads on SRA as well, so he pulled some of them down. Can we assemble those? And turns out to be not too hard. So you click on one of these. So this is something he ran a little while ago, and this shows you the execution of that WDL. Now this was a couple weeks ago, on February 13th, and as you can see, here are the 5 main steps. That last step is kind of a placeholder step. And here's how long it took. It took 5 minutes to run step 1. It took 2 minutes to run step 3 or 2. There was a little bit of gap where nothing was happening between, and then by the end of it all, it was all done. You see this kind of like big summary page of, so I guess if it isn't clear, the WDL describes, and this is true for the CWL Nextflow too, it describes the pipeline is kind of generic state. And it's up to the user to populate it with inputs, specifically input files, right? So this is about assembling a viral genome. Okay, well then give me the reads, right? So that's like the most basic input. But it's the same WDL you run on every single sample, every single project. It's just as each time you run it, you're populating with different inputs. What's beautiful about the language, or any of these languages, is the way you talk about those inputs might be totally specific to your compute infrastructure. So on Google Cloud, you'll have like GS bucket URIs, right? On DNA Nexus, you might have these opaque file objects. On the cluster, you'll have like a Unix file path on the NFS file system. It doesn't care, right? The language basically says, when you write that script in the command section of the WDL of the task, you can assume everything's in Unix at that point, because basically it did all the work of taking that file from wherever it is, on the cloud or wherever, and pulling it down to the computer so that when you're actually running the code in that script, which is in your Docker container, you can assume those files are right there in front of you on your local path. Does that make sense? So anyway, this kind of shows you what it looked like to run this, and here's what all the inputs were for all the different steps, and here's what all the outputs were. Some of the outputs are files to some of these steps. Some of them are actually numbers or strings. Those are useful too, and so on and so forth. Now, in this case, I said we grabbed some reads off of SRA and asked ourselves, can we assemble a genome? One of the quickest visual ways of looking at that, there's, the actual final output. This is just me knowing my code, by the way, nothing to do with WDL itself, but like the output that you're gonna care about is this FASTA file, that's the assembly, but kind of a visual output is just like, here's the coverage plot. So if I were to ask myself how successful was this assembly, I might look at something like this. So this is a 30 KB genome. Here's what my coverage looked like. Here's the result of that assembly process. I'm going to leave this up here because, and this looks like assembly coverage plots you've probably seen before. There's some gaps and missing stuff and whatever, and you can interpret that. What I wanted to do now is also flip over to showing you what this exact same thing looks like in the Broad's Terra platform, which is all based on Google. So the project you just saw in DNA Nexus, that was all executing in Amazon. And here I've got the same little workflow here, the assemble de novo with deplete. The interface looks a little different. When you set up the launcher, it shows you all of the file and non-file inputs all at once, and so asks you to fill them in, tells you which ones are required, which ones are optional. You can do some file selectors, fill it in, that kind of stuff, to then run the analysis once you've done that. And once you kick it off and run the analysis, it comes over here to the job history. You can see I've done this a few times. Sometimes it succeeds, sometimes it doesn't. And here's here's the time I ran, I think, the same file. Let's see if it is. And similarly, when you look at the Terra execute. And again, it's the same WDL. It's talking about files differently, but I gave it the same input file. So on the Terra side, you're giving it these Google bucket strings, and on the DNA Nexus side, you're talking about these like DNA Nexus file IDs. And again, that's the thing that should change. If you ran Cromwell on your cluster, the way that you run it is, you run this Cromwell executable, and you submit to it the WDL you want to run and a JSON file that describes the inputs that you want to bind. And those inputs are going to be like NFS file paths to like that's where my file is. So, and similarly, this also gives you a little timing diagram of what it looked like to run. There's some more colors here because it's kind of telling you the difference between, step 1 was spin up a VM, step 2 was pull down the Docker image, which actually takes a while because it's a heavy Docker image. Localization is copy files from the bucket to my VM, and this pink part is the actual compute, right? So you can kind of visually see the efficiency of this, and it's not terribly efficient if you're just doing a single workflow. It becomes very efficient though if you're talking about like a large throughput of I want to do this on a couple hundred samples or that kind of thing. And similarly, when you look at the outputs, you might say, okay, what does the final output look like, and where's the coverage plot? So here, what it does is it drops you straight onto the Google bucket because Terra itself is just a thin layer over Google. At least that's the way it's currently implemented. Sorry, I'll try to zoom in on this a little bit because I realize this is kind of small. But what that means is, what's kind of interesting is the way you pay for Terra. You don't pay the Broad. So for DNA Nexus, you pay DNA Nexus to use it, and then they pay Amazon. But for Terra, you don't pay the Broad anything. You actually just set up a billing account with Google and then link it into Terra, and then you just pay Google for whatever your compute and storage is. The Broad is not in and there at all in terms of financial transaction. But what I wanted to kind of show is that the resulting plot that you get out of here, ideally, am I looking at the same file? Should, even though it was computed separately, should be the same. Yep. This is 3401. 341. Yeah. So it should be the same. In fact, when I looked at this before, a lot of the numbers and metrics end up being the same. So this is kind of what I mean by portability. I can run it here, I can run it there. And I'm not really going to show, just for the sake of time, what it looks like to run on the command line. That's something that I think hopefully Joel and Andrew can kind of demonstrate at some point as well. And they've been kicking around a lot of different other pipelines in this space. But that's basically kind of the dream. And I'm just going to cut a little bit to the end. I do have like just, let's see. There's more I could say just about the trends and kind of the ways that these types of directions have been going. There's been a lot of effort around interoperability and defining APIs for how to do this kind of stuff. While everyone has a very different infrastructure setup, can we talk about the same pipelines? Can we pass them around in the same way? And what I'm kind of ultimately trying to do is, be able to have pipelines that we can all share on something like DocStore. I've mentioned this before, but this is kind of a standard tool repository service where if you look at their site, they've got WDLs, they've got CWLs, they've got Nextflows, and with each of those, and you browse them around, they actually have these little buttons like launch this thing on DNA Nexus or launch it on Terra, or download the thing and run it on your cluster. That's kind of, in my mind, what the ultimate kind of portability scenario would be. So yeah, I talked more than I wanted to. I actually really want to hear people's kind of questions and experience on this, but yeah, sorry about that.
No, thank you for that. I think also showing it running on 2 different platforms really gets home to the point of the portability of things. DNA, and I know we did start a little bit late. I don't know what your time schedule looks like, but we can open up for discussion.
Yeah.
Okay, great. We'll leave it open for.
Yeah.
We'll leave it open for the next 10 or 15 minutes if folks have questions.
I have a question.
Yep.
So I've been playing with Nextflow for the last couple of weeks, and I think you already kind of highlighted one of the big major differences is the way that WDL and CWL both have these tasks that are outlined kind of separately, and then you have your workflow, which is kind of described in its own separate file. And when you're kind of demonstrating the code, one of the things that I noticed is that in your task file, you have certain runtime parameters, like how many CPUs to use and like what kind of Docker container to use. And that was one of the interesting things that I liked about Nextflow, is the fact that you can create a config file and have that import other config files. And you can actually define your runtime parameters for each step in the process differently, depending on where you want to run it. So for example, if I wanted to run it using AWS, I could specify much larger CPU requirements than if I just wanted to run the pipeline locally. And I was kind of curious to hear if, one, what was your perspective of having those tasks be separate files? Do you think that helps with readability and accessibility? And two, do you think that, is there more flexibility in how you define your runtime parameters?
Yeah, so I think those are two separate things, so I'll hit them separately. The separating into different tasks and stuff is not necessary at all. It's just something that I've done because I thought it was useful for me to be able to reuse stuff. So I could have several different workflows that glue together tasks in a different way. And I don't want to like rewrite all that. So I just kind of, I thought it was easier to organize it in that kind of way. But that's not required at all. You don't have to do the imports. You don't have to do any of that kind of stuff. On your other question about the runtimes and how easy it is to separate the configuration, so there's two pieces of this. One is the thought that perhaps to the extent that the hardware requirements of a task are just dependent on the task, then maybe they should be coded at the task level. To the extent that they're kind of dependent on aspects of the data, so people do this kind of thing where maybe there's some sort of function that's performed on the data, then maybe you want more memory or less memory if you have a lot of data or a little data. So I've seen that type of thing done too, where this is replaced with a variable. But then to your point, sometimes the optimal hardware to run on is different depending on your platform. And so there's two ways to do a WDL runtime. Memory and CPU are the most generic kind of catch-all things, but there are ways to layer on overrides on top of each other. And some of these things, for example, this DX instance type is ignored by Terra and Cromwell, but is only for DNA Nexus. This is a code that only means something to that platform. And so this gets used for that platform. And for other platforms, some combination of this gets used. And the optimal cost-effective VM for 15 gigs of RAM and 8 CPUs might be different for Amazon and Google, so it kind of calculates that differently. The other thing you can do is have infrastructure-specific config files that set defaults, right? And you can define the direction of who overrides what. So that's in there if you wanted to. So yeah.
Thanks, that's really helpful.
Hey, Danny, I have a question. Thank you for the awesome presentation. It really helped explain what WDL and Cromwell and all this is. I'm just curious about the development aspect when you're writing WDLs. Do you typically work on like just a local machine first, test things there, then port it to the cloud and test? I mean, how often do you see something not, works locally and it doesn't work in the cloud, things like that?
Great question. Usually if it works, if it's containerized well, the only reason things should fail in one versus the other is if the resource requirements are not possible in one or the other. I think that's a discussion that's perhaps even longer. I have, well, I had a slide here, but I have a CI setup under Travis where basically every time we push a commit to change the WDLs, it'll do a few different checks. It'll do a syntax kind of validation. So WDL comes with like just a very simple validate. I'm not gonna try to run anything, but is everything kind of parsed correctly? And then it will do some kind of end-to-end tests on some really tiny bits of data. So I obviously don't do this for all my WDLs, but for some of them, I found like a really tiny piece of data that like I can run through this in a short period of time. And so I have that running on Travis too. you know, if you don't have an automated setup, you know, yeah, you can test it on your local computer. You can test it on a cloud setup. Cromwell can do that. You know, so I use Cromwell to run things real quick on Travis VMs. So Travis VMs are pretty tiny. So if it runs there, it probably runs anywhere. The CZI folks use MiniWhittle now, which is a bit more lightweight. It doesn't parallelize all the different backends. It just runs on one machine.
Cool, thank you.
I am curious to hear, I know some folks have kind of talked about what they've, but I'm kind of curious to hear what else, if there are other folks who've kind of dabbled in this a bit more, or played with their favorite languages or anything.
I know we've got Robert on the line here. I don't mean to put you out there, but I know Robert's been using a lot of Nextflow for Staphopia, Bactopia, and everything that they've got going down at Emory. I don't know if you want to speak a bit on how you made that decision, Robert. I know actually I've asked you for next month's call to kind of also give that insight, but I think this is also appropriate time for that conversation to happen. Oh, no mic. So Dan, if you could see the chat there.
Yeah, okay. That's, I mean, maybe next time. I would love to hear more. I'll just say that, I mean, because people have asked me, like, you know, why should you use one or the other? I mean, I have my reasons why I chose what I chose, and it actually is not because it's out of the same institution as me. I mean, I think there are reasons I do like WDL in particular. That being said, if you're using any of these three, I'm pretty happy. I think it's not too hard if you've written your stuff in one of them to either hand port or write a parser or whatever, like to actually, if you look in docstore.org, you'll see a number of pipelines actually presented in multiple of these languages because they're the most similar to each other. It's a lot harder to get a snakemate pipeline into this than it is to convert these three to each other. If you're starting from scratch, I would nudge towards WDL or CWL mostly because they tend to, just because of the ecosystems they're in and the standard bodies that kind of thrive around them, they seem to be a little bit more first class in that area. But you know, there's also lots of, I mean, especially if you're starting from a place where you're porting in a lot of existing pipelines from Nextflow, it makes a lot of sense to be there too. I think I've openly wondered how long it's going to take before Nextflow really kind of joins in the GA4GH effort. But at that point, I think a lot of this won't even matter. I think for me, I kind of mentioned before, part of, if you're starting from scratch, part of it is like defining which language ecosystem you want to be able to share pipelines with. And also just keeping an eye on portability, making sure you can execute other people's stuff and that type of thing. It's, in my ideal world, common infrastructure, like lab infrastructure, common infrastructure, you know, whether it's your lab's compute setup, whether it's, you know, the AMD portal or like an Ames cloud or something like that, would all have some layer that could execute one of these languages, such that people could just share their pipelines on Dockstore and then we'll just pull it out and just have a place to run stuff. So that's kind of the world I hope for.
Danny, I had a question about that portability of WDL workflows across different compute infrastructures and how things like that get configured. For example, you showed how we can use Terra and then we have access to those Google resources. But for example, if I wanted to run your workflow here in Virginia and I have AWS resources, how do I encode things like authentication and the compute EC2 resources versus Google Cloud resources in the way that you described how the DNA Nexus has specific instance names or image names rather? Yeah.
Well, describing the hardware resources isn't too bad because at the very minimal, some of the common language like CPU and mem are universal. And so the right instance to choose kind of gets chosen by the scheduler based on what backend you're running on. But obviously, the authentication, so each of these different modes kind of has documentation. And Joel and Andrew are going to have the most kind of stories around like getting this up and running. There's definitely activation energy, right? It's going to take a while to get something like that running on your setup, you know, at first. But once you do, it just opens a world of like being able. But a lot of it is just setting up the config file, right? Having it point to the projects, having the right, you know, in the case of Google, having the right service account tokens, in the case of AWS, having the right IAM like kind of parameters set up and that type of stuff, and link to whatever you've got set up there. But once that's set up, once it goes, it's kind of cool to just see it go.
All right.
Yeah,
Thank you.
I can definitely add to that and say that, you know, there was a little bit of a learning curve of trying to learn how to code in like the Nextflow and Groovy and everything like that. But honestly, like 99% of my time has been trying to set up the AWS backend to accept everything. So.
Have you gotten that to work?
Yeah, it works great.
Awesome. And then once you're there, it goes, right?
Yeah, the trickiest part was figuring out permissions because all of the tutorials and guides online are out of date. So.
Hard agree with that one. Yep, absolutely.
That's partly, and we've seen this across the board, that's partly because it's a quickly moving target for many. Like the right way to do something in AWS or Google changes every year sometimes. So, and the point of this is to add the layer where, you know, if it changes every time, you just upgrade your version of Cromwell or whatever, but hopefully your pipeline code won't change, right? It's all that other details getting abstracted away.
All right. So any other questions for Danny? Well, I'll keep it open for the next couple minutes here. All right. If not,
I don't have a question.
Ahead,
just a quick comment. Kelsey summed it up pretty well. I was talking to her about playing with Nextflow, and she's like, "Yeah, I drank the Kool-Aid. It's pretty cool to see it work." And I too, I've just barely played with Nextflow. I mean, just the most minimal workflow ever, but it was very cool to see it run locally, and then it almost instantaneously run on our cluster here. It's the portability is amazing with these workflow languages.
Yeah, and once you see like portability, like kind of for the first time, like in a real tangible way, and then once you see like parallelization on a large data set, it's quite impressive. So.
Yeah, I think that's actually something, Danny, you kind of just went over really quickly, but that ability to scatter and then gather, I think that's actually what I was most impressed about whenever Joel was showing me some of the workflows he was putting together in Salt Lake City. So thank you for that. Thank you, everybody. I'll be able to post this recording on StaPH-B.org afterwards. Thank you, Danny. That was fantastic. I'll be keeping everyone up to date on the next StaPH-B talk via email. Thanks, guys.
Thanks.
Thank you, Danny.
So, Danny, before you take off, I was curious, have you seen or